Okay, I guess we started recording, so I can start the introduction. Today, we're pleased to have Mustafa Ümit Öner from National University of Singapore, and he will be talking about histopathology. I wanted to look up what histopathology means. Maybe there's an introduction of that in the talk, but it's basically the study of living tissue at the microscopic scale. Mustafa will be talking about the application of deep learning and image processing techniques on these microscopic images, which may be huge given the imaging technology of the day. So let me introduce him quickly. He got his BS in 2013 and his master's in 2016, both from the electrical engineering department of METU. He's a PhD student in the computer science department at the National University of Singapore. And he's interested in developing novel machine learning models and machine learning based information systems for digital histopathology and integrative multiomics to support diagnosis and therapeutic decision-making in cancer. He is focusing on the intersection of digital histopathology and genomics in cancer research. And specifically, his current research is to develop novel machine learning models to predict genomics features from digital histopathology images. So I will leave the floor to Mustafa Ümit Öner.
Yeah, thanks for the nice introduction. I think we can directly jump into the details of the work. Today, I'm going to share with you some part of my PhD work. Specifically, today's title is How Strong Are the Weak Labels in Digital Histopathology? I will give some brief background and then we will jump into the details of my work. I want to start with the clonal evolution of cancer and tumor formation process. Actually, cancer is defined as the uncontrolled growth of abnormal cells. It starts in a single cell or in a small group of cells that acquire some mutations and gain survival advantages over the other cells. This group of abnormal cells is called as clone, and their uncontrolled growth is called as clonal expansion. During this clonal expansion process, some further mutations occur and new genetically different cell clusters or subclones appear within the tumor microenvironment. This process is called as genetic diversification. However, all of these subclones cannot survive within tumor microenvironment because there are some selective pressures. Only the ones that can escape from these selective pressures can survive within the tumor microenvironment. And this process is called as clonal selection. Overall, this reiterated process of clonal expansion, genetic diversification, and clonal selection goes on and results in a heterogeneous tumor structure. Here you see nodes with different colors. Actually, each node represents a cell cluster or a subclone with different phenotypic features or biological capabilities, such as cellular morphology, gene expression, or metastatic potential. As the number of these subclusters increases, actually tumor becomes more and more heterogeneous. This is what makes cancer a highly deadly disease. In fact, in 2019, it is estimated that there were around 9.3 million cancer deaths. And in the fight against the cancer, our primary diagnostic tool in the clinic is the histopathology. A sample is collected from the patient, and then this sample is processed and glass slides are prepared from the sample. Finally, pathologists analyze these samples under the microscope and make a diagnosis. However, there are some challenges in this process or in general in histopathology. First of them is the increased workload. It is estimated that there will be around 30 million new cancer cases by 2040. Another factor contributing to this increased workload is the decreasing number of actively working pathologists. Another challenge is inter-observer variability. What is inter-observer variability? If we want to define inter-observer variability, we can define as this. If we give the same slide to two pathologists, it is possible that two pathologists can come up with different diagnoses or different results. This is called as inter-observer variability. In fact, both of these factors, the increased workload and inter-observer variability, directly affecting the patient outcome. Okay, these are the challenges, but some good things also happening. First of the good things is the development of digital slide scanners. Actually, development of digital slide scanners transformed histopathology into digital. Now we can scan the glass slides and store them as whole slide images in digital media. This enables us to get remote consultation and diagnosis. This is especially valuable if we don't have pathologists in our hospital, or if we have inter-observer variability. We can resolve inter-observer variability by getting a third view from anywhere from the world. Transformation of histopathology into digital, in a way, is our lives, but it's not eliminating all of the challenges. Still, these slides are read by the pathologist. Only this time, they are reading these slides through digital screens instead of under microscope. So their workload is still high. Another good thing happened in the machine learning domain. Thanks to the availability of big data and computation power, actually deep learning models achieved huge success in computer vision tasks in different domains. One of the domains actually is the digital histopathology. Here I got a few example tasks from digital histopathology. For example, metastasis detection, mitosis detection, mutation prediction, or survival prediction. Some of the examples that deep learning models already shown to perform quite good. In fact, we can combine these two technologies, the digital slide scanners and the deep learning, to support pathologists in diagnostic and therapeutic decision making, to reduce their workload, and more importantly, we can improve the patient outcome. Okay, we can combine these two technologies, but these also has some challenges. It's not straightforward. One of the challenges is we have huge images. A whole slide image is around 100,000 by 100,000 pixels. So there is no way we can process this whole slide image at once in traditional deep learning models. Another challenge is these deep learning models require a lot of labeled data, but most of the time in the clinic, these whole slide images are not annotated at all. I want to try to explain this, why they are not annotated in the clinic. Here you see a whole slide image at the left side. At the highest resolution, it is around 95,000 by 70,000 pixels. At the right side, you see a higher resolution version over this whole slide image. Actually, this region corresponds to this tiny green square on the whole slide image. This is just 2000 by 2000 pixel at the highest resolution, and it corresponds to this tiny green region over the whole slide image. If we ask a pathologist to annotate this slide, the pathologist need to go through such high resolution images and draw the boundaries or contours between different regions within the images. As you see, it is really a tedious and time-consuming task. It's almost not possible. Okay. Although we do not have pixel level annotations or manual annotations by pathologists, we have one quite valuable thing. We can easily obtain sample level or slide level labels from digital or the electronic health records, from pathology reports, or even by using some different data modalities. We call this kind of slide level or sample level labels as weak labels. For example, we can get and read the pathology report. We can learn whether a slide contains cancer or not, but we don't know actually where the cancer resides within the whole slide image. We call these type of labels as weak labels, and they are quite valuable for us to exploit. Okay, in today's talk, we are going to discuss the novel MIL models that we developed to address these two main challenges in digital histopathology. First, we want to tackle huge images, gigantic images, and then we want to utilize the weak labels. We are doing this by using the multiple instance learning paradigm. We are representing a whole slide image as a bag of patches cropped over the whole slide image. And all of these patches now can be processed by traditional deep learning models. And then we are using the sample level or slide level labels as our bag labels. And then using the bag and bag label pairs, we are training our MIL models end to end. In fact, we developed novel MIL models and show their usefulness in different tasks at the intersection of histopathology and genomics. Today, I'm going to share with you two of these works. In the first one, I will try to show you how we can achieve digital histopathology tasks only by using weak labels. In fact, this work is published in ICLR 2020. And then we will go into the details of how we can predict the percentage of cancer cells or the tumor purity from digital histopathology slides using a MIL model. This work is currently under review in a journal. Let's start with the first work, which is weakly supervised clustering by exploiting unique class count. By the way, if you have any questions, please feel free to interrupt at any point. Okay, you don't have to wait until the end of presentation. In this work, our primary research question, given only the image and image level weak labels, can we obtain some fine level information about the image? For example, given that image and the piece of information that the image is a mixture of cancer and normal cells, can we obtain the corresponding segmentation mask showing the cancerous and normal regions within the image? In order to achieve this task, we are representing the input image as a bag of patches, and then we are using the number of cell types within the image as our bag label. By using the bag and bag labels, we are training our MIL model. Finally, we are using the trained MIL model to segment out the cancerous and normal regions within the images. We are going to see the details of this block diagram actually. But in order to achieve this task, we are introducing a novel multiple instance learning task. First, I want to introduce this task to you. This task is based on a bag label called unique class count. It is the number of unique classes among all instances inside a bag. For example, here you see 3 example bags. In the first bag, we have patches containing only cancer cells. So there is only one type of cell, which is cancer cell. So the unique class count is 1. So the label is UCC1. Similarly, the second bag contains the patches of only normal cells. So again, it also has the label of UCC1. On the other hand, in the last bag, you see both the patches of cancer cells and the normal cells. So there are 2 types of cells. So the corresponding bag label is UCC2. After introducing the bag label, now we can introduce the task. Task is unique class count prediction. Given a bag of instances, we want to predict the corresponding UCC label. And we want to do this by training a MIL model. The important thing in this point, we don't know the labels of individual patches within the bags. We only know the corresponding UCC label to train our models. In fact,
I have
this
a
is
question.
Yeah, sure.
I have a question here. So in a pathology image, you may have different types of regions. So cancerous and normal could be one possibility, but you may have other regions like necrosis or lymphocytes. So is it necessary for your model to get that information as a prior information?
No, we just group them based on the whether it is metastasis or cancer or the other cells. Actually, I just mentioned as normal, but any other thing is just fall into the other group. First group is the
your bags are labeled with UCC1, UCC1, or UCC2.
Yep.
So is it possible to make it UCC3 or UCC2 only corresponds to the heterogeneous back? So
Yeah,
how
you
does
can,
it work?
you can, if you have enough number of components from other groups as well, for example, fat tissue, necrosis regions,
Mhm.
you can label them as UCC2, UCC3, UCC4. It's not necessary to have only UCC1 and UCC2 labels. In fact, we showed that for other, for a broader range, it's also working.
Okay, thank you.
Yeah, you're welcome. In this task, actually, we don't know the labels of the patches. In fact, this is our ultimate objective. Given bags of instances and corresponding back level UCC labels, we want to assign labels to individual instances inside the bags. We are proposing a two-stage framework for that. In the first stage, we are training a UCC model. This is the task. This is the model to achieve the task that I have introduced in the previous slide. And then in the second stage, we are using this model to cluster the instances within the bags. In the first stage, the UCC model get a bag of patches as input, and then by using the bag and bag pairs, we are training this UCC model. Once we train the UCC model, we are using the model as a feature extractor, extract the features of all the patches inside all the bags, and then we conduct a clustering to assign labels to the individual instances. Here, the idea is very simple, very basic idea. If a model can learn how to count number of clusters within a bag, actually it inherently learn how to identify individual clusters as well. The idea is as simple as that. Okay, let's go into details of these two stages one by one and then see what's happening. First stage is the unique class count model. At the input, we have a bag of patches, and then our first module is a deep neural network extracting the features of the patches inside the bag. This module gets the first patch and obtain a feature vector, get the second patch, obtain another feature vector, third patch, another feature vector. Once it obtains feature vectors for all the patches within the bag, we are summarizing these feature vectors, extracted feature vectors, into a bag level representation by using a MIL pooling filter in here. In our case, this is a novel distribution pooling filter based on kernel density estimation. Once we obtain the distribution of the extracted, estimate the distribution of the extracted features, the second neural network use these estimated distributions to predict the label of the bag. Here is in this architecture, novel part is the MIL pooling filter in here. Instead of using traditional point estimate based pooling filter like mean pooling and max pooling, we are using a distribution pooling filter to estimate the distribution of the features because we want to pass as much information as possible to the next stage. If we use mean pooling or max pooling, actually we are losing information. The advantage of using distribution pooling, it is more expressive compared to the point estimate based pooling filters. In fact, this is a completely separate paper, which is under review for now, but we are not going to go into the details of this work for today. But this was the novel part in this architecture. Okay, once we construct this pipeline, this model is trained end-to-end by using bag and bag label pairs. After training this model, we are using the trained feature extractor to extract the features of individual instances. And then over the extracted features, we are performing a clustering to assign labels to the instances. As we have seen, our ultimate objective is assigning labels to the individual instances. So our ultimate performance metric is clustering accuracy in this task. But as obvious, since this is a two-stage framework, the ultimate performance metric, which is clustering accuracy, also affected by the performance of the first stage, which is the UCC model's performance. Here you see this
Mustafa,
relationship. Yep.
Mustafa, how did you define the loss function on
Here,
the
this is
back
just
labels?
This is for UCC1 and UCC2. This is binary classification, and this the loss function includes the categorical loss.
Okay.
Categorical cross entropy loss is calculated as by using categorical cross entropy.
Okay.
In fact, there is another branch in this architecture which I skipped. There is an autoencoder in here as well, which includes a L2 loss. But for simplicity, I just skip for this talk.
Okay.
In
So
the original,
the loss is
yep.
The loss is defined on the classes of UC1 and UC2.
True.
It's just binary classification. Okay.
Mm-hmm. Yeah, in the original one, as I said, there is another loss component, but we can skip for now.
Okay.
As you see, the clustering accuracy is affected by UCC accuracy. In the y-axis, you see the clustering accuracy. In x-axis, you see the UCC accuracy. As expected, as the UCC accuracy increases, the clustering accuracy increases as well. At the limit, if we have a perfect UCC model, this study proves that we can perfectly cluster the individual instances inside the bags without knowing the labels of individual instances during training. After theoretically proving that it can work, we also checked the performance of this framework on a real-world breast cancer metastasis segmentation task. Metastasis segmentation in breast cancer is a crucial step in cancer staging, actually, and we are aiming to solve this problem by using our weakly supervised framework. We are given the patches and corresponding UCC labels. As we discussed, the labels is just based on how many different kinds of cells included within the image. If the image contains only normal cells or metastasis cells, the label is UCC1. Otherwise, it is UCC2.
Well, Mustafa,
a
question about the previous slide. What is the number of clusters in the unsupervised clustering case? The K.
We are using the UCC number as our number of clusters. We are using K-means clustering here. In this task, we have UCC1 and UCC2, right? In the clustering phase, we are using 2 means clustering, for example. If we have
I see.
UCC
So
numbers.
in this plot over there, you have 2 classes in both of the axes. UCC has 2 classes and clustering also over 2.
Yeah, true. Since we already given the UCC labels, we can use this information during clustering as well.
Okay, thank you.
Yeah, okay. Okay, given the images and corresponding UCC labels, we want to find the corresponding segmentation masks. We are using our weakly supervised clustering framework such that we represent the image as a bag of patches, and then we use the labels and the bags to train our UCC model. Using the trained feature extractor, we extract the features of the patches and then do the K-means clustering to obtain segmentation mask. To compare the performance of our framework, we are also training a fully supervised UNet model, which is a famous biomedical image segmentation model. The main difference between the UNet and our framework, UNet requires manual annotations during training of the model, which is not the case for our framework. Here you see a few example patches from our images from our test set. The columns correspond to the test set images. The first row show the original input images. Second row show the manual annotations, and the rest shows predictions from different models. The third row, the predictions from our framework. Fourth row is the predictions of the UNet model. And then finally, we see in the last row, the predictions obtained by K-means clustering over the raw pixel values as a baseline performance. If you look at the predictions of K-means, it's not performing well. The predictions are not consistent with the ground truth. But on the other hand, if you compare the predictions of our UCC framework and UNet model with the ground truth masks, they are giving quite good results. We are also comparing these methods quantitatively. Here you see the details of the data set. And then we are using the pixel level statistics on the test set as our performance metrics. You see true positive rate, false positive rate, true negative and false negative rate. Finally, the pixel accuracy in the columns, and then rows correspond to the different methods. If you look at the k-means in the last row as an unsupervised baseline, the pixel accuracy is around 0.5, which is like a random performance. But if you compare the performance of our weakly supervised framework to the performance of the fully supervised UNet model, you see that it approximates the performance of the fully supervised UNet model. In fact, this shows us we can achieve digital histopathology tasks only using weak labels as well if we formulate them correctly. This was the first part of the presentation.
Mustafa, I have one more question.
Yeah,
So,
sure.
did you check how the system behaved for other types of tissues? So suppose that, so here it either takes the bags of cancerous patches, normal patches, and a heterogeneous mixture of normal and cancerous patches. But that could be your assumption, but did you check what would happen if you use a heterogeneous patch containing, for example, fat tissue and normal tissue, or fat tissue and cancerous tissue? So did you check such kind of cases?
In these slides, we have some fatty regions as well. As I said in the beginning, we treat the cancerous regions or metastasis regions as one class, and all the other normal cells, all the fatty regions, necrosis as another group. So in the clustering as well, all the fatty regions and necrosis regions also goes as normal regions. This was for breast cancer lymph node segmentation. We checked the same model on lung cancer intratumor heterogeneity prediction as well. We tried to predict the number of subclones within the tissue. This number of subclone information labels obtained from genomic sequencing. We use this information and then train the model and do the training of the model. You can use on other tissue types with heterogeneous structures as well, but it depends how you formulate the problem.
Okay, thank you.
You're welcome. I think we can move into the details of the second work, which is predicting the tumor purity from histopathology slides using a MIL model with distribution pooling. Let's start with the definition of tumor purity. It is defined as the percentage of cancer cells within a sample, and it is used for sample selection for genomic sequencing. A sample is collected from a patient, and then this sample cut into multiple portions. And from the top and bottom of each portion, the glass slides are prepared. And then pathologists estimate the tumor purity over these slides. By looking at these estimations, the portion with the highest tumor purity sent for genomic sequencing. This is really important because the sequencing methods or molecular tests require a minimum percentage of cancer cells within the sample to reliably detect the genetic abnormalities. Otherwise, you can miss some of the mutations or the changes, genetic changes within the sample. So it is crucial to estimate the tumor purity correctly during sample selection for genomic sequencing. As I said, the tumor purity over the slides are estimated by the pathologist, but actually how do they estimate? Given a slide, pathologists choose some representative regions, and then at high resolution, they go and count the tumor nuclei and the normal nuclei over these high resolution images. As you see, this is really tedious and time-consuming task, and it involves really high inter-observer variability. Another problem is that the pathologist estimates are not consistent with the genomic tumor purity values, which are inferred from sequencing data and accepted as accurate for downstream genomic analysis. Here you see genomic tumor purity versus pathologist estimates. If you look at the graph, you see that pathologists usually overestimate the tumor purity, which is a really a big problem, because you might have false negatives in your sequencing analysis. To support pathologists in sample selection for genomic sequencing, we proposed an MIL model predicting the tumor purity by using the slides prepared from the sample. A sample is cut into portions, right, as we discussed. One of the portions is sent for sequencing, and 2 slides are prepared from the top and bottom of this portion. We are using these 2 slides to represent that sample. Actually, we are representing the sample as a bag of patches cropped over these 2 slides, and then a feature extractor extracts the features, distribution pooling filter summarize the extracted features into a back level representation by estimating the feature distributions. Finally, another representation transformation module predict the tumor purity by processing the estimated distributions. During end-to-end training of this model, we are using the genomic tumor purity values as our back labels, so we can train an accurate model. We check the performance of this model in 8 different cohorts in the cancer genome atlas for 8 different cancer, and a local lung cancer cohort in Singapore as well. In each cohort, except the Singapore cohort, there were around at least 400 patients, and we segregate the patients into training, validation, and test set at patient level with the ratios of 60%, 20%, and 20%. We train our model on the data of the training set patients, and then we choose the best set of model weights based on the validation set performance. Finally, we check the performance of our model on the data of unseen patients in the test set. During performance evaluation, for each sample in the test set, we created 100 bags and then obtained tumor purity predictions from the trained MIL model. Finally, we get the average of these 100 predictions and use this value as our sample prediction. We use correlation coefficient and absolute error as our performance metrics. For one cohort, you see 2 scatter plots in here. In the first one, you see the genomic tumor purity versus our MIL model's predictions. And then in the second one, genomic tumor purity versus pathologist estimates. If you look at the graphs, the MIL models are giving much better performance than the pathologist estimates. This was for one cohort. What is the situation for all the cohorts? Here you see both of our performance metrics, the correlation coefficient and the mean absolute error. In the y-axis, correlation coefficient. In the x-axis, you'll see the mean absolute error. Different colors represent different cohorts. Triangles shows the performance of our MIL models. Circles represents the performance of pathologist estimates. If we have a really good model, we expect this model to appear around the top left corner of this graph, which means it is giving high correlation and low mean absolute error. In fact, if we look at the graph, we see that all the triangles are concentrated around the top left corner of the graph, which shows us the MIL models outperforming the pathologist estimates. After showing that we can correctly predict the tumor purity, we go one step further and we generate tumor purity maps showing the variation of tumor purity within the slides. This is especially valuable for low purity samples, which requires to cut out the high tumor content regions and send this region for sequencing. This process is called as microdissection. In this step, we are aiming to automate this microdissection process by producing tumor purity maps. By looking at the tumor purity map, we can detect the high tumor content region and we directly cut out this region and send for sequencing. Another good thing about these tumor purity maps, we can better understand the tumor microenvironment by analyzing the interaction of the normal regions and the cancer regions. How we obtain these tumor purity maps? For each region, for each 1 millimeter square area over the whole slide image, we are creating a bag of patches, and then we obtain the corresponding tumor purity prediction from the trained MIL model and assign this value to the corresponding location in the tumor purity map. Once we obtain all the predictions for all 1 millimeter square area in the whole slide image, we end up with a proper full tumor purity map. Here you see two examples from the same cancer type. One is from TCGA cohort and one is from Singapore cohort. If you look at the Singapore cohort example, the pinkish one is showing the digital histopathology slide, and the colorful one showing the tumor purity map. In the dark region in the pink image correspond to the tumor region. Actually, it is just all the cells invading the spaces in the lung tissue. And if you look at the tumor purity map, it is represented with yellowish and greenish color, which correspond to the high tumor purity values. So it correctly shows us where the tumor is. So we can do a microdissection over this region and do the genomic sequencing. In fact, this was the last slide for my talk for today. I tried to show you we can achieve digital histopathology tasks even only with weak labels, and then we see a MIL model can predict tumor purity from digital histopathology slides. Here you see the list of people contributing this work, make it possible. I want to thank all of them, my supervisors, Week 1 and Ken, TAC members, our collaborators, Bioinformatics Institute, NUS, and finally my lab mates in BII. And lastly, I want to thank to all of you for your time and attention. If you have any questions, more than welcome. Please feel free to ask any questions, any comments, any suggestions. I'm open to anything. Thank you.
Okay, thanks, Mustafa. So thanks for this great talk. So it's a really nice work, and personally, I enjoyed a lot since it is closely related to my research area. So thanks again. So any questions from the audience?
I have a question.
Sure.
Does selecting the patches require any supervision? For example, what happens if I get a patch that has more than one label?
For the first part you are asking, I guess.
Yes.
Okay. This is really a good question, actually. All the machine learning guys consider all the problems as IO problems. You give some input, you obtain some output. In fact, the reality is different. As you mentioned, if you select a patch at the boundary, your patch can contain both cancers and normal cells. And if this is the case for majority of your patches, you end up with a big problem. But in this case, while you are designing your architecture, you need to use the domain knowledge. You need to work with the medical doctors. If you choose these patches so big, obviously all of your patches will contain some normal and some cancer cells. But if you choose your patch size such that you are making sure that you are including enough number of cells, but you are not including so many cells so that your patches are all UCC2. You need to rely on the domain knowledge of the experts at this stage. It is the case for all medical problems. So we have to work with the medical doctors and the experts from different domains while designing our architectures, our models, formulating our models. I'm not sure whether this answers your question.
Yes, thank you. I guess it becomes even more complicated when we have more than 2 labels.
Yeah, true. We design artificial problems by using MNIST and CIFAR data sets. In this case, in MNIST and CIFAR, you have 10 classes, right? If you mix the classes and create artificial bags in the original paper, we have such analysis. Once you increase the number of classes in the bag, you need to also increase the number of patches in your bags, because if one of the classes is minority, so small, let's say you have 10 classes, right? each of your bag, let's say, consisting of 100 patches. If you have only one instance of a class, it's highly likely that your model will miss this example. You may require to have at least some balance between the classes in your bag, so your model does not miss anything. You're
So
right.
it almost becomes like an egg chicken problem, right? Again, we need some supervision from pathologists, I guess.
Yeah, you
Maybe not
definitely you need.
Yes.
Yeah, true.
Thank you.
Yeah, you're welcome.
As a follow-up question, so did you check the effects of having different patch distributions in a bag? So you may have 20% normal patches versus 8% cancerous ones, or 50-50. So did you find a chance to check the effects of
for
having
each
different
patch,
distributions?
yeah, we check the ratio of the metastasis patches within the images. We create our training, validation, and test set such that it has a normal distribution around 0.5. They have some outliers, but I think we filter out 20% in both sides. At least one class is represented with 20% of the patches.
Okay. More questions? More questions from the other audience?
Yeah, any suggestions are welcome. It will help to improve
Okay.
our work.
I will be happy to talk with you after the talk. So
Yeah,
it's
sure.
actually very exciting and nice work. Yeah.
Yeah, thanks a lot.
More questions? Okay, so that's the end of the talk. Thanks for coming, and thank you, Mustafa, for this great presentation. And
Thanks for
hope to
inviting
see
me.
you, all of you, in our next meetings.
Okay,
Okay.
thanks a lot.